and the beta linkage. Alpha linkage, it is formed by alpha types of alpha sugar. They form alpha linkage. Beta linkage is between beta type of sugar. Here we don't have alpha and beta kinds of glycosidic linkage. There is no anomeric carbon here. There is simple glycosidic linkage formation between N9 and carbon number 1 of the sugar, that is called glycosidic linkage. Okay, this one is a nucleoside. It is formed as a result of sugar addition on the nitrogen base, adenine, guanine, cytosine, thymine, and uracil. They form their respective nucleoside as a result of this sugar addition. Prokaryotic cell, they have extra chromosomal DNA. They have single chromosome, but this single chromosome it is circular in structure. It is found simply suspended in the cytoplasm in the form of nucleoid. You know this one. There is a single chromosome in the bacteria cell even. The other one, non-chromosomal DNA in bacteria, it is found in the form of plasmid. We have extra chromosomal or non-chromosomal DNA in the bacteria cell. It is found in the form of plasmid. We human beings, we have this extra chromosomal in the mitochondria, and plant cell, they have extra chromosomal DNA in the chloroplast. RNA, this one usually is present in the nucleus in cytoplasm. In the nucleus, RNA it is found as a result of transcription reaction. To have this information, for example, this DNA it is always found in the nucleus. So as a result of transcription reaction, transcription means copying DNA information into RNA. RNA is messenger RNA. It is formed by this transcription reaction in the nucleus. So we have RNA molecule in the nucleus. This will be translocated into cytoplasm for protein synthesis. There is a protein synthesis. Protein will be synthesized by using the information found in the messenger RNA. Protein is synthesized in the cytoplasm. So we have RNA molecule in the nucleus as well as in the cytoplasm. The other example for this RNA molecule in the nucleus is ribosomal RNA. Ribosomal RNA, it is synthesized in the nucleolus of the cell. Nucleus contain chromosomal DNA, and nucleolus is the dark region found in the center of nucleus, used for the synthesis of ribosomal RNA. We have ribosomal RNA in the nucleolus. So what is the function of this ribosomal RNA? Who can tell me functions of ribosomal RNA? Messenger RNA, it will be used for carrying the information, or it will copy the information found in the DNA and used for protein synthesis, or guide amino acid sequencing in the protein structure using nucleotide sequence in the messenger RNA. Ribosomes, yeah, they are components of ribosome. Good. They make ribosome with protein, ribonucleoprotein. It is formed in the ribosome and used for protein synthesis. Okay. So RNA it is present in the nucleus as well as in the cytoplasm. DNA it is always found in the nucleus, and the extra chromosomal DNA it is found in the mitochondria and chloroplast. Nucleic acid, it is a weakly acidic substance. It is weakly acidic substance and rich in phosphate molecule. There are phosphates in the DNA molecule or RNA molecule. They have this phosphate group plus the sugar it is found. So they are made of nitrogen bases, sugar, and phosphorus. Both DNA and RNA, they are a polymer of nucleotides. There is polymerization of nucleotide using this phosphodiester linkage for the formation of larger DNA and RNA. We call them polynucleotides. So they are joined together by a phosphodiester linkage. This linkage, it is formed between the 3' OH group of the first nucleotide. This one is by the first nucleotide reacted with 5' OH of the second nucleotide. So this is from first nucleotide attached with 5' OH of the second nucleotide. Okay, it is formed by using the phosphates. We'll see this one later using this diagram. by using diagram. We'll discuss this one later. So it is formed between the 3' OH of the first nucleotide and 5' OH of OH or phosphate. We can say it is phosphate. This one is not hydroxyl group, but it is phosphate found at 5' carbon of the second nucleotide, and they form this phosphodiester linkage. So this is a typical structure of a polynucleotide, or it is segments here. There is leftover structure here from this direction upward, and the other one here also we have a leftover structure. This is a segment of a polynucleotide. Now let us see how this phosphodiester linkage is formed. The first nucleotide, it contains 3 components, as we have seen before. Nucleotide contains 3 things: nitrogen base, sugar, and phosphate. There is phosphate here attached at carbon number 5. This one is carbon number 1, carbon number 2, 3, 4, and 5 here. This one is carbon number 5. Numbering here, it is given with prime: 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. The number in the nitrogen base, it is given without prime. We have seen this one. Numbering for this nitrogen base, it is given without prime. The carbon here, they are labeled with prime: 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime is the element found in the sugar units. So the sugar bind base and phosphates. This base, it is attached at carbon number 1 of the sugar. The phosphate, it is attached at carbon number 5 of the sugar. This is one nucleotide. So how these nucleotides are chained together for the formation of a polynucleotide, it is by using a phosphodiester linkage. Phosphodiester linkage, it is formed by 3' OH. The 3' OH group here, 3' OH group of the first nucleotide react to this 5' phosphate group. The 5' phosphate group plus the 3' OH group of the first nucleotide, they form a phosphodiester linkage. This is phosphodiester linkage. Typical phosphodiester linkage, it is formed by the 3' OH group of the first and the phosphate 5' carbon of the sugar. The whole strand, if you can see the whole strand, this one, the direction, it have direction first, and then the direction is run from 5' free end or unreacted 5' end of the first nucleotide and reacted 3' OH group of the last nucleotide. So this is run from 5' end to 3' end. Okay. This is the direction of nucleic acid. Nucleotide sequence. Sequence of a polynucleotide is known as primary structure. Okay, primary structure, it is unmodified structure of nucleic acid, or it is after immediate synthesis of DNA or RNA molecule without further modification. We have a primary structure. This primary structure of nucleic acid contain nucleotide sequences. Simple nucleotide sequences are they are a primary structure of nucleic acid. We call them a polynucleotide. They are chained together by this phosphodiester linkage. So what is the direction here? Polynucleotide chains, they are found in 5' to 3' direction. They are found in this direction. So this is taken as a convention between different scholars. It have direction, it is run from 5' to 3' direction. But the linkage between nucleotide, it is run from 3' to 5' direction. The linkage found between the nucleotide is run from 5' to 3' direction. For example, this is one phosphodiester linkage. It is run from 3' end to 5' end. The whole length, it is run from 5' end to 3' end. Okay, this is consensus sequence for this nucleic acid. So we have seen this one. Each polynucleotide chain has 2 ends. 5' end unreacted phosphate on the left hand, 3' end carrying unreacted hydroxyl group on the right hand side. So these are the 2 ends of our polynucleotide. it is running from 5' into 3' end. Last time, yesterday, we have seen the sequence of amino acid in the protein structure. They run from N terminus to C terminus. Like this, polynucleotide have this direction. It is run from 5' to 3' direction. So this is for primary structure of DNA and RNA. Primary structure means it is unmodified structure or without a double-strand structure. It contains a single-stranded DNA and RNA polynucleotide. It is running from 5' to 3' direction. That is primary structure of DNA and RNA. It exists in the single-stranded form. That is DNA and RNA. RNA, it is always found in single-stranded structure, but DNA is found in the double-stranded structure. That means the primary structure of DNA, it will be modified or converted to a secondary level of DNA structure. There is a double helix formation. Okay. So therefore, in DNA and RNA, the letter here they represent nitrogen bases found in the DNA and RNA. For example, A represent adenine, guanine represent G represent guanine, cytosine. It is represented by C. Thymine, it is represented by T. These are the nitrogen bases found in the DNA and RNA molecule. The sugar found in this DNA and RNA. In DNA, we have a deoxyribose sugar. RNA contains ribose sugar. The RNA, it is ribose sugar. So if a polynucleotide is segment of DNA, we have this deoxyribose sugar. If the segment is RNA segment, we have ribose sugar as a pentose sugar. When these are hydrolyzed, they produce nitrogen bases, sugar, and phosphate. We know this. Nucleotide hydrolysis, or when these are degraded, they form nitrogen base, sugar, and phosphate. The sugar, it is important either for synthesis of another nucleotide, or they will be used for energy. There is energy production by this ribose sugar metabolism. Okay, ribose sugar, who can tell me ribose sugar, how this ribose sugar is synthesized in the glycolysis pathway? Is there anyone who can? Hello? Okay. Ribose sugar, it can be synthesized through HMP pathway. There is one pathway in the glycolysis pathway, HMP, that means hexose monophosphate pathway. There is conversion of glucose to ribose. Glucose, it is a 6-carbon sugar. Ribose, it is a 5-carbon sugar. So this one is formed in the HMP pathway. As a result, it can be used for nucleotide synthesis. Plus ribose, it can be used for glucose synthesis. So this HMP pathway. So this sugar, after hydrolyzing this nucleotide, it can be used for synthesis of another nucleotide, or it can be oxidized into carbon dioxide and water for the formation of energy. The phosphate, it is, again, it will be used for synthesis of another nucleotide. But nitrogen base, it is degraded for the formation of urea. It contains nitrogen. Nitrogen, you know, it is toxic in the free form. So urea is formed by this nitrogen base degradation. Nucleotide, they contain two types of nitrogen base, purine and pyrimidine, pyrimidine bases. Purine, it is a structure that these are adenine and guanine. Pyrimidine, it is cytosine, thymine, and uracil. You know this. How purine nucleosides are attached with sugar. The sugar, it is attached nitrogen base by using N9 or nitrogen number 9. Pyrimidine, it is attached with the sugar unit by using N1 of the pyrimidine ring. Nitrogen number 1 of pyrimidine ring reacted with carbon number 1 of the sugar. Nucleotides, they are a phosphorylated form of nucleoside. nucleosides, when they are phosphorylated, they form a nucleotide. So nucleotide, it can be a diphosphate, it can be in the single phosphorylator or double phosphorylation or triple phosphorylation. There are three types of nucleotide: mononucleotides, dinucleotide, or triphosphate nucleotides. Here is the linkage found between bases and the sugar. It is formed by this glycosidic linkage. So purine and purine nitrogen base and the sugar, they are attached together by using this glycosidic linkage. It is formed between 1' of the sugar and 9 of this purine nitrogen bases. Pyrimidine nitrogen bases, they are using this N1 of N1 nitrogen number 1 of this pyrimidine nitrogen base reacted with carbon number 1 of the sugar. They form 1,1 glycosidic linkage. This is a glycosidic linkage found between the sugar and nitrogen base. Phosphate, they are attached at carbon number 5' of the sugar used for this phosphorylation. This phosphate, it can be 1 or 2 or 3. This is a nucleotide structure: purine deoxyribonucleotide and pyrimidine deoxyribonucleotide. They are formed by this phosphorylation reaction of nucleoside. Let us see the structure of DNA. DNA, it is formed by a polymerization of deoxyribonucleotide attached together by this phosphodiester linkage. There is a phosphodiester linkage which chaining the single deoxyribonucleotide into this deoxyribonucleic acid, which is a polynucleotide. Each deoxyribonucleotide or nucleoside, they contain nitrogen base and sugar, deoxyribo sugar, it is found. The nitrogen bases are purine and pyrimidine bases. The two purine nitrogen bases are adenine and guanine. The three pyrimidine bases are cytosine, thymine, and uracil. These are the three pyrimidine bases. Primary structure of DNA is a single-stranded DNA or linear sequences of deoxyribonucleotide linked by this phosphodiester linkage between the 3' OH group of the first nucleotide and 5' of the phosphate nucleotide on the second nucleotide. This is the direct. It is formed between 3' OH group of the first nucleotide and 5' phosphate group of the second nucleotide. By this phosphodiester linkage, there is a polynucleotide or a deoxyribonucleotide formation in the linear DNA structure, or in the primary. We found this linear single-stranded polynucleotides. The sugar phosphate backbone. Sugar and phosphate, they form the backbone of these linear nucleotide sequences. The variable or bases, they are found variably inside of this backbone. This is one strand. Okay, it is running from 5' end to 3' end. The backbone, it is made by sugar, phosphates alternatively. There are sugar and phosphate alternatively. The nitrogen bases, they are found inside of this backbone by this right angle triangle. For example, this is one strand. This is one strand, and the bases, they are hanging from this inside, from this strand. So we have this backbone. It is made by sugar phosphates backbone, and the bases, they form the strand. They form the strand and found inside of this backbone. Okay, nucleotide polymer has free phosphate group at 5' position of the sugar and a free 3' OH group on the final nucleotide. Okay, final nucleotide contains 3' OH group on the last nucleotide. So by this, the sequence of the single-stranded polynucleotide, it is written in this direction, 5' to 3' direction. So the bases, they are abbreviated abbreviated, and they are found inside of this backbone. We have seen this one by this diagram. The backbone or the outside structure, it is made by sugar and phosphates, and the inside structure, it is made by alternative or variable nitrogen bases. For example, this one is guanine, cytosine, adenine, thymine. Alternatively, these nitrogen bases, they are hanging inside from this backbone of primary structure of DNA. This is not the natural structure when DNA is found in double-stranded. That means the primary structure of DNA will be converted to secondary structure by base pair formation. There is a base pair formation between nitrogen bases found in this single-stranded structure of DNA molecule. The secondary structure of DNA, this one, it is formed by two strands of DNA. There are two strands of DNA. They are paired together. There is pairing the two strands. So the two strands in the secondary structure of DNA, they are found in opposite direction. That means they are found in anti-parallel direction. One of the strand, it is run from 5' to 3' direction, and the other strand, it is running from 3' to 5' direction. So this is one typical example for this anti-parallel or opposite in direction immunome. Typical example for this DNA structure is running from 5' to 3' direction, one of the strand, and the other strand is run from 3' to 5' direction. And bases, they are stuck inside of the two strand. This is because of their polarity. They are non-polar or hydrophobic. So they hide themselves from water facing. They fear water, therefore they hide themselves inside of this double-strand structure. Sugar and phosphate, they are polar or hydrophilic, therefore they face outward toward the water molecule because of their polarity. So the sugar phosphate backbone is found on the outside structure. Bases, they are stuck inside of these two strands of DNA molecule. In the RNA molecule, we have a single-strand structure, single-stranded RNA molecule. They have a single strand. So bases, they are found inside. Sugar phosphate, they are found outside. There is no possibility of avoiding water contact in the RNA molecule because of single-strandedness in the RNA molecule. So bases of one strand paired with bases of the other strand, there is base pair formation between the two strands. This makes the DNA molecule is complementary. There is complementarity between the two strands of DNA molecule. One strand of DNA, it is complement to the other strands of DNA. It is due to base pair formation. So bases of one strand paired with the other, the next respective base of the other strand. Adenine, it is always paired with thymine with two hydrogen bond. Guanine paired with adenine here is paired with thymine by two hydrogen bond. Guanine, it is always paired with cytosine with three hydrogen bond. There are three hydrogen bond between guanine and cytosine. So which one is more strong? Which one to have more strong attachment? It is guanine and cytosine. GC, rich region of DNA, it is more strongly attached or paired together than the AT-rich region of DNA molecule. During transcription or replication of DNA, it is always occur at area where AT-rich region is occur. AT-rich region, AT means adenine, thymine, base pairing, it is occur by two hydrogen bond. So the linkage here, it is weaker than GC-rich region. Therefore, transcription and replication of DNA, it is started or initiated here at AT is rich in the DNA molecule. The negatively charged phosphate group in the sugar unit, they expose themselves to the outside structure of this double-strand DNA structure. The two strands of DNA, they are coiling around an imaginary single axis. They coiled around this imaginary axis. and they form a right-handed double helix. Right-handed double helix. It is rotating double-strand DNA around this axis and form a double helix. So the plane of sugar, we have sugar here, phosphate and sugar backbone on the outside structure, and bases they are found on the right angle. They are found in the right angle for this DNA molecule. The plane of sugar are at right angle for right angle of that of this nitrogen bases. The helical structure of DNA, it is length about 10 nucleotide residue. There are 10 nucleotide residue lengths per one helical turn. This is the length of one helical turn. Per one helical turn, we found 10 nucleotide residue on this DNA molecule. So the helical structure of DNA it is discovered by Watson and Crick. These are the two scientists. They discovered the structure of DNA and developed this model of DNA. From this model of DNA, we have a B form DNA. There are three forms of DNA, by the way: the A form, B form, and Z form DNA. Including C form DNA. There is also C form of DNA. The A form DNA is formed during dehydration or in alcohol solution. For example, the A form DNA is formed. A form DNA is shorter in helical length. The helical turn in the A form it is shorter than the B form. B form it is the most stable structure of DNA at physiological condition. That means the pH, the temperature on this physiological condition, the B form DNA is more stable than the A form and the Z form. The A form it is formed during dehydration and it have shorter lengths of one helical turn. Z form, this one is completely different from the A and B. This one it have left-handed helical turn. A and B they have the same right-handed helical turn, but the length of one helical turn is different between A and B. This one it contains 10 nucleotide residue, but the A form it contains 11 nucleotide residue per one helical turn. The Z form it is left-handed, plus it is more shorter. It contains 12 nucleotide residue. The Z form DNA and C form DNA it is found in the mitochondrial DNA. Mitochondrial DNA it is circular. It look like prokaryotic DNA. This one it is circular DNA. Our DNA in the nucleus it is linear. It have two ends. There are two ends. These two ends, there are telomeres at the two ends of our DNA. Telomere, it is the end of our DNA. Telomere is found. Okay. In between, we have this coding region, or the information is coded between of these two telomeres. In the mitochondrial DNA, it is circular. It have circular DNA, like it is look like prokaryotic DNA. Using this information, there is one hypothesis. Mitochondrial DNA, it is assumed as a prokaryotic, and it is engulfed prokaryotic cell. From this eukaryotic cell, they engulf prokaryotic cell and they form this mitochondrial DNA. So this one, it is a derivative, or it is derived from prokaryotic DNA, and it have similar DNA structure, which is look like prokaryotic DNA. So they form Z form DNA, which is left-handed helical turn and plus shorter helical turn. C form DNA is found also in the mitochondrial DNA. This one it can be formed during replication or transcription reaction. We form Z form DNA and C form DNA. Okay, these are the three different types of DNA structure. The first one, the A. This A, it shows you double helical strands of DNA coiling around the central axis of this double-stranded DNA structure. Per one helical turn, it can length up to 34 angstrom length. So it have 10 nucleotide base pair formation between per one helical turn. We have this 10 nucleotide base pair formation. Between two strands, there is 20 angstrom distance between the two strands. During this helical turn formation or coiling around the central axis, there is depression formation, or groove formation. There are two types of major groove formation, major and minor groove. The major groove, it is more deeper than minor groove. So this groove, it is important to bind protein. When it is important to bind regulatory proteins. As a result, they regulate the expression of DNA. DNA is it is regulated whenever it is activated. There is expression of DNA into RNA. Whenever it is inhibited or inactivated, there is inhibition of transcription or expression of DNA. It is expressed. So protein, they are attached on this major groove and minor groove for the regulation of this DNA expression. The B form DNA, the B structure, this one, it shows you the sugar phosphate backbone here. We have this alternative sugar phosphate backbone, and bases, they are attached together by this hydrogen bond. For example, we have thiamine attached to this pair bases, this adenine molecule by this hydrogen bond. Between guanine and cytosine, there are 3 hydrogen bond formation. This is it will be repeated, or it will be occur based on the type of nucleotide. So if you know the sequence of one of the strand of DNA, and then automatically we can draw, or we can estimate the other strand nucleotide sequences or base sequence. Base sequence can be written based on the sequence of the first strand. So they are complement to each other. They are complementary between each other. Thiamine, if there is thiamine from this strand, there is adenine on the other strand. If there is guanine from this strand, there is cytosine from the other strand. This is base pair formation, or the space here, it is only fit for guanine. Then guanine paired with thiamine, guanine paired with cytosine, using these 3 hydrogen bond. So this one is stronger than 80 base pair formation. The third structure, this one, it shows us the backbone, sugar phosphate backbone, and the base inside. These bases, they are inside because of their polarity. They are hydrophobic, therefore they hide inside from feeding this water. They hide themselves from avoiding water contact. The sugar phosphate backbone, they are found on the outside structure because of their polarity. They face toward the water environment. Coming to this RNA structure, RNA is single-stranded, except there are exceptional RNA molecules, but most they are found in a primary structure. They are found in the primary structure or in a single-stranded structure. The single unit of RNA molecule, it is ribonucleotide. It is made by a ribonucleotide, but RNA is by deoxyribonucleotide. Here, the sugar in the RNA molecule contain oxygen at carbon number 2 of the sugar. Pentose sugar contain oxygen at carbon number 2. It is hydroxylated at carbon number 2 of the sugar. Uracil is only found in the RNA. Thiamine is found in the DNA. We have seen this one. The difference between uracil and thiamine, as I have told you before, uracil is non-methylated, or it is formed from this one will be used for thiamine synthesis by methylation reaction. So this is methylated form of uracil at carbon number 5. Carbon number 5 of uracil is methylated. The nuclear DNA is secondary structure, but RNA is found in the primary structure with exceptional T transfer RNA molecule. It is found in the tertiary level of nucleic acid structure. After transcription reaction, there is modification into tertiary level of nucleotide structure or nucleic acid structure. That is true for this transfer RNA. We'll see later the structure of transfer RNA. It will be discussed later. Okay, this is